when the polypeptide chains run parallel and are held together by hydrogen and disulfide bonds, then fiber-like structure is formed. By altering the temperature or pH or any other chemical or physical factors, we can denature the protein. That means its biological activity or its nature is being disturbed. That is called as denaturation of protein. Only maltose is the enzyme that can be used for the conversion of maltose to glucose. No other enzymes can be used. Hello everyone, this is Amli Unnikrishnan from the Department of Chemistry. So today I'm back with session 4 of the chapter Biomolecules. So in the last session, we started off with polysaccharides, right? And then we moved on to the importance of carbohydrates, as well as we started off with amino acids and the different classification of amino acid based on different factors, as well as the Zwitter ion form of amino acids was discussed in the last session. So in today's session, we are going to start off with the next biomolecule, which is very important, which is very familiar to you also, that is proteins. Right, and we are going to discuss the structure of proteins and what is denaturation of proteins, which is very important in the examination point of view, as well as enzymes. Clear? So let's begin with proteins. Okay. So when we were discussing regarding amino acids, that time itself I have mentioned that amino acids are the building blocks of proteins, right? So let's see. Proteins are the polymers of alpha amino acids. Okay. So alpha amino acids will combine together to form proteins. So which is the basic unit of proteins? It is alpha amino acids, and they are connected to each other by peptide bond or peptide linkage. So it is necessary to remember what is the type of bond present in proteins. Okay, it is peptide bond or peptide linkage, which is formed between amino acids. So each amino acids will be bound together by peptide linkage to form what? Proteins. A polypeptide with more than 100 amino acid residues having molecular mass higher than 10,000 is called a protein. So many amino acids will be joined together by the peptide linkage to form proteins. So proteins is classified into two types based on their molecular shape. So based on their molecular shape, proteins are classified into fibrous and globular. So let's understand what is fibrous proteins. When the polypeptide chains run parallel and are held together by hydrogen and disulfide bonds, then fiber-like structure is formed. Okay. So in fibrous proteins, it will be like fibrous structures and they run parallel to each other. So it will be like fibers and they are bound together by hydrogen bonds or disulfide bonds, right? So they are called as fibrous. They look like fibers, right? Now this structure results when the chain of polypeptides curl around to give a spherical shape. So from here itself you can see globular. So it will be curling around instead of running parallel to each other. This is parallel to each other, right? So now what happens is if they are in a curled structure, that is in a round or spherical structure, we call it as globular protein. So based on the shape in which they occur, we can classify it into fibrous or globular. Such proteins are generally insoluble in water. So fibrous proteins are insoluble in water. Globular proteins are soluble in water. Now fibrous example are keratin which is present in our hair, right? So you might have heard keratin treatment which is done for hair, right? Yes. And myosin which is present in muscles. Right. Now examples of globular protein is insulin and albumin. So insulin also familiar to you, right? So this classification is very important in the examination point of view as well. So what are fibrous and what are globular proteins, and the differentiation between it is important. So moving on to the structure of protein. Okay, so in detail, let us study about the structure of protein. So we have understood the classification, that is fibrous and globular. Now we are going to study about the structure of protein, how each of these amino acids is linked together and the final protein structure is formed. Okay, so the first one is a primary structure. Okay, primary structure. The primary structure of protein refers to the specific sequence. Okay, it's a specific sequence in which various amino acids are present in it. That is the sequence of linkages between amino acids in a polypeptide chain. If this sequence changes, then the nature and the function of protein changes. So you can see that there is a specific order in which each of these amino acids has to be linked. So you can see these round structures here. Each of these are different amino acids. So it has to be ordered or has to be forming bonds in this particular order itself, then only we can say that it is this particular protein. So if by any chance any of this order is changed, the nature of the protein will change and the function of the protein will also change. So this primary structure is very important, which cannot be altered. If it is altered, the nature and the function of the protein will change. So this is the primary structure. Moving on to the next one, that is secondary structure of the protein. Okay, so it refers to the shape in which a long polypeptide chain can exist. This structure is resulted due to the regular folding of long peptide chain. So a regular folding of a long peptide chain will happen and you will get the secondary structure. So primary structure, you can see it is a simple structure. Now this primary structure will start curling or coiling around to form a secondary structure. Folding will take place. This folding is caused due to hydrogen bonding between hydrogen atom of NH2 group and oxygen atom of the CO group of different amide, same or different polypeptide chain, and the chain exists in two different forms. Either it can be alpha helical structure or else beta pleated structure. So the first one you can see is alpha helical structure. So this you can see, it is the primary chain which you can see. Now what happens is, we have studied about the linkage, right? It is NH-CO linkage which is between the amino acids. While we were discussing amino acids, we have discussed. So now you can see here, this is one chain here, another chain here, you can see that. So now what happens is, extra linkages will be formed between each of these chains. So that is hydrogen bonds which is being formed. So because of this extra hydrogen bonds being formed, what is going to happen? The primary structure will curl around to form the secondary structure. So it can be in two forms. One is alpha helix. So this is alpha helical structure. You can see it's like helix. Right? Okay. So this is horizontally shown. If it is vertically shown, you can see it's a helical structure. So that is why it is called as alpha helix. Right? Now you have beta pleated structure also. So beta pleated structure. So if you take a piece of paper and start folding it one above the another, and if you open it, what you can see is a pleated structure will be obtained. So the same way you can see a beta pleated structure here. So here also, this is one strand, you can see another strand and another strand. Now what happens is, there is again hydrogen bonds which is formed between each of these strands. So that is the beta pleated structure. So because of this hydrogen bonds, again it is curling around and this is the secondary structure. Now moving on to the tertiary structure, refers to the overall folding of the polypeptide chain, that is further folding of two structures. Two shapes are fibrous proteins and globular proteins. So this is how you are obtaining fibrous and globular protein. So primary structure will form extra hydrogen bonds and form secondary structure. Now secondary structure further will curl around and form the basic structure of the protein, that it can be globular or fibrous. Right? Now secondary and tertiary structure are stabilized by hydrogen bonding, disulfide linkages, van der Waals forces and electrostatic forces. So we have already discussed this. Primary structure will be there. Then how this secondary or tertiary structure is formed, how this curling will take place and how the shape is getting changed. It is because of the formation of hydrogen bonds, disulfide linkages, van der Waals force and electrostatic force. Right. Now the last one is the quaternary structure. So before moving on to the quaternary structure, you can see the tertiary structure here. Again from the secondary structure, again it is going to curl and form the tertiary structure. Now moving on to the last one, that is quaternary structure of protein. Some of the proteins are composed of two or more polypeptide chains referred to as subunits. The spatial arrangement of these subunits with respect to each other is known as the quaternary structure of protein. So quaternary structure is the final structure of the protein. Again, this tertiary structure. So what does that mean, quaternary structure? So you can see this is one unit or one subunit, and further another tertiary structures will be there, right? So this is one tertiary structure. Again, further other tertiary structures will be there which will combine together and curl together to form the quaternary structure. So you can see this is one subunit, another subunit, another subunit, another subunit, right? All of this will combine together to form the quaternary structure of the protein. So I hope it is clear. Primary structure, secondary structure, tertiary and quaternary structure. So now after studying all these four structures of protein, it is important to remember that the primary structure of the protein is the very, very important one, because if that gets altered or if that gets changed, the whole function and nature of the protein will be changed. Okay. So if you have to change or alter the function of the protein. You can keep changing the primary, secondary, tertiary. So that is important to remember. Moving on to the next one, that is denaturation of protein. So this is very important. Okay. Now, what are native proteins? Let us see. The protein found in a biological system with a unique three-dimensional structure and biological activity is called as a native protein. So native protein, it will have its own function, what it is supposed to do in our body, right? Each protein has its own function. Right. So it is having its own biological activity. It has a proper function and a proper nature. Now, what is denaturation of protein? So from here itself you can say denaturation. We are going to alter the nature or function of the protein. So how it can be done? Let's see. The laws of biological activity of proteins. When a protein in its native form is subjected to physical changes like change in temperature or chemical change like change in pH, this is called as denaturation of protein. So in its own state with its particular function and biological activity, that protein is called as a native protein. So by altering the temperature or pH or any other chemical or physical factors, we can denature the protein. That means its biological activity or its nature is being disturbed. That is called as denaturation of protein. Example is coagulation of egg white on boiling. So while boiling the egg, we can see that the egg gets harder, right? It is coagulation which is happening. That is actually a denaturation of protein is happening. So the native protein is not like that, right? The original egg is not like that. Once we start boiling, what is happening? Its nature is getting changed. Right, it is becoming hard. Same way, curdling of milk also. All these are example of denaturation of protein. Right, now moving on to the next one, that is enzymes. Okay, so enzymes are proteins that catalyze biological activities. So what are enzymes? They are catalysts which helps in the reactions of biological reactions. They are very specific in nature and catalyze only particular reaction for a particular substrate. So enzymes are very particular in their nature, that is they can be used only for a particular reaction and for a particular substrate. So if the substrate changes, the enzyme used should be also changed. So enzymes are usually named after the particular substrate or class of substrate and sometimes after the particular reaction. For example, the enzyme used to catalyze the hydrolysis of maltose into glucose is named as maltase. So which is a substrate here? It is maltose. Maltose is getting hydrolyzed. So maltose is a substrate. So maltose will give you glucose, that is 2 molecules of glucose only when this particular enzyme is present, which is that enzyme maltase. So the maltase name comes from the substrate which is maltose itself. Okay, so only maltose is the enzyme that can be used for the conversion of maltose to glucose. No other enzymes can be used. So enzymes are used to catalyze biological reactions and enzymes are very, very specific in nature. So you can't use, for example, invertase is another enzyme which helps in the hydrolysis of invert sugar. Okay, that is sucrose. So invertase can't be used here. Only maltase can be used in the hydrolysis of maltose. Alright. Again, the enzymes used to catalyze the oxidation of one substrate with the simultaneous reduction of another substrate are named as oxidoreductase enzyme. So what are oxidoreductase enzyme? Which helps in the biological reaction which is undergoing both oxidation and reduction. That is why they are called as oxidoreductase enzyme. So in the next session, we are going to discuss about hormones and few other important topics by which we will come to an end to the chapter. So I hope the concepts that I've covered in this session is clear for you. So that's all for today. Thank you.